Okay, so if
Okay.
you are ready, I'll start.
Okay, so yes.
Okay, correct. So good morning, afternoon, evening, hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you for joining us the 35th seminar and Imperial College London Day. I started my crazy 10 weeks of seminar trips with the third stop being University College London, UK, where I gave my talk 6 hours ago. In other words, I am joining SynBYSS from London, and actually one of my pioneer speaker today, Professor Baldwin, is also from London, but another university, Imperial College London, the rival school, I guess, and which is located again, in London as well. And actually, I'm now giving my talk. I will give my talk in Imperial College in July, I think after 6 weeks from today. And so I'm talking at 4 p.m. rather than 10 a.m. my time in St. Louis with the Professor Baldwin from London. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, again, the Professor Baldwin. He is co-director of the Imperial College Center for Synthetic Biology and director of the EPSRC Center for Doctoral Training in Biodesign Engineering. As you know, he does not need introduction, but I just want to say one thing. He is a true leader and pioneer leading UK or European synthetic biology community. So this is my absolute honor to be with him today. So I'm really thank so much for your time and support for the SynBio community. And Professor Baldwin, podium is all yours now. Thank you so much.
Thank you, Taesok, and it's a real pleasure to contribute to your seminar series, which is, really amazing to bring the world community together with a particular focus on young researchers. So I will share my screen. Okay, so can you see my slides okay? Yeah, great. Okay, so I just wanted to give a bit of an overview about the Center for Synthetic Biology here at Imperial College, and also some information about our doctoral training center as well, which may be of interest to young people out there wanting to start their career in synthetic biology. So, this is kind of an approximate timeline of kind of major events that have been going on. At the center itself was founded in 2009 by Paul Fremont and Dick Kitney. So synthetic biology, though, actually kind of predates this somewhat. So this was the first EPSRC funded center in the UK in synthetic biology. But the first foray into SynBio, came through iGEM, and Paul and Dick led the first iGEM team from Imperial College. I think that was probably about 2006. And I sort of joined the year after in 2007 and sort of started getting involved in synthetic biology. And I think there's a really interesting lesson and reflection here in terms of, the role of iGEM and young people in really, the great burgeoning success of synthetic biology as a field. And that's something that's been really important to us here at Imperial College, because on the one hand, we had kind of, leadership and people who kind of took on iGEM, but then also we had this sort of bottom up, iGEM was a real beacon for some of our best students to come into SynBio. And then pretty quickly in around 2008 and 2009, we developed the first undergraduate module in synthetic biology. So that is a final year module in the life sciences department, but it's also co-taught with bioengineering, and it still is to this day. And it's, I think, the only cross-faculty undergraduate program that I know of at Imperial College. So that was, fairly groundbreaking, and it's really nice that we've been able to continue that. But what that does is it really acts as a beacon. I've had a number of students over the years who said it was when they did the undergraduate SynBio course that they knew what they wanted to do, so so that, acting as a beacon for young people. And I think what stimulates people is, I spent 10 years or more doing, biochemical research, which was really kind of like discovery based. It was focused on observing, understanding how biological systems work. And the key difference with SynBio is with our undergraduate students, it's the final course that they take of their degree. And we kind of say to them, well, look, we've given you all of this knowledge over 3 years. Now we're saying over to you. Think about how you can use this knowledge to solve problems, to do something creative. And once students kind of get the bit on that, then it's really quite transformative for a lot of people. And they kind of go, wow, okay, this really opens up what I want to do. And I think that's been underlying the success of iGEM as well. It's that mentality that it brings to the field. So for us, to come back to my timeline, that sort of training pipeline that we've developed of the undergraduate module, those people who are excited by it, they kind of go on to do our MRes. And then, it was great in 2019 that I led this successful bid for a doctoral training center in bioengineering, that we now have a training program that sort of takes students forward into doctoral training. And so that continuation of the pipeline of training has been really important. And those people, go on to, become postdocs in our labs. They go and found startup companies. They go and become, lab PIs. So, we're mature enough now that we've seen our students kind of go into all of those different destinations. And that's really amazing. And I think that underpinning is just so important to actually, really kind of bring this field forward and enables, the creativity and enthusiasm of all of those amazing students to contribute to the field. And the other most recent activity we have is now we just recently got an AI for Engineering Biology Consortium funded here at Imperial, where we are kind of really trying to focus on that interface of applying AI approaches to SynBio is the next interface and the next challenge for engineering biology. So the center itself, we've got the largest center of synthetic biologists, I think definitely in the UK, probably in Europe. And, we cover really a very diverse set of spaces, from microbial, mammalian, in vitro protobiology, and, the underpinning control engineering basis of that. And so there's a really fantastic interdisciplinary group of people here at Imperial doing world-leading research. And so our structure of it, we have our center, which focuses on the education. And then we also have here, based at Imperial, SynBYSS, who are interacting with lots of SMEs and kind of really trying to support that translation and industrialization space. And we're also very fortunate to have the London DNA Foundry, which then also helps support those activities. And we all work with industrial partners as well. So academic groups work with industrial partners, as well as them kind of working, with the Foundry and SynBYSS. So we have sort of coupled with, the training and education pipeline, this translation pipeline as well. So just to tell you also a little bit about the Doctoral Training Center. So that started in 2019. And really we came to that with, thinking, well, SynBio was founded on the interface of engineering and biology. And so, that was kind of really key, but we recognized that as it's developed and the workflows that underpin synthetic biology, that we really needed to add a third leg to the training stool by bringing in data science. So, SynBio is and should be a data-rich subject. And so, the Biodesign Engineering Doctoral Training Center was formed on the basis of trying to bring students forward on all three of these aspects, which we believe are really important to underpin the discipline. And it was founded really, the quote here from Marcus Cacheta, who's a CSO of SynThase, one of the sort of big startup companies in SynBio in the UK. And he said, what SynBio needs is, in terms of PhDs, is lab competent coders and code competent biologists with a firm grasp of entrepreneurship and other business development skills. And that strapline has really become kind of the framing goal of our doctoral training center that we are trying to live up to. So we have, this center is also in collaboration with UCL and University of Manchester. So we have students across all three of those institutes, but we also bring those students together to train them. And the pipeline is everyone does the first year is the master's course at Imperial College with foundation training in synthetic biology and a long project, which then feeds into their PhD years. But we also have training across all of those cohorts that runs across all 4 years of the program as well. And one of the things we've done through this is we've brought automation training into the course as well. So we set up what we call a student biofoundry. So we've got 6 OpenTrons robots, and so that the students do a training module where they learn how to code. All of their coding is done in Python, and the OpenTrons robots are coded in Python. So everything kind of integrates kind of across the training that we're doing. And that's a really fantastic part. Because they get trained on this before they go into their research labs, that really kind of sets them up for thinking about how best can we use these new types of approaches in order to help take things forward. And this year we did our first 1 week kind of a retreat with the students, where we had a week-long entrepreneurship training, which was a fantastic week where we kind of got really good training. And just as an example, Zach Sorrells here, kind of really world-class pitch training event that I wish I'd been on 10 years ago. Honestly, it was really superb. So, working with industry is a key part of what we do. And we also established a system where we really try and work with micro and small companies as well, which are a key part of the ecosystem for synthetic biology. It's not all big companies. And so being able to work with small companies and find mechanisms why, we can give them cheap access into co-supervising PhD students, give them, a way into being allied with world-class research groups and facilities is a really important part of what we do as well. So we've currently, we've got 3 cohorts in place. We've just recruited 15 students who are starting this October. And we have 1 more cohort that will be starting in 2023. And to all those undergraduates out there, we can now accept a small number of overseas students into the program as well. So overseas students are eligible to apply. It is competitive, obviously, but it's kind of really great to be able to now do that, to be able to kind of reach out and consider people from all over the world who are interested in synthetic biology. So, if you're looking for, training, if you're at that stage of your career, then a shameless plug, but please think of Imperial College. And that's it from me, Taesok. Thank you.
Thank you so much. I mean, the absolutely amazing talk and also introduction to one of the best university for the synthetic biology. And also I completely agree with you and young people's excitement actually, in the past, drove our growth and of course, UK's growth in synthetic biology. And also UK has been encouraging young people to join the field and also Imperial College, making Imperial, as I said earlier, become the hub of synthetic biology worldwide. And also very interestingly, you just said you collaborate with the UCL and the Manchester, and actually I gave my talk today in the morning at UCL, and then going to give my another talk at Manchester tomorrow, and that's kind of fascinating coincidence to me.
But you're going to all the best places. What can I say?
Yeah, but the UK too many great universities, so I cannot miss any of them. So that's absolutely my honor
I
to
think
be
that
there.
there's also a message for academics in that presentation as well, right? So so basically the message is, if you want to have good postdocs, you've got to have the training pipeline to bring your students through. If you want to grow synthetic biology at your institution, you need to find mechanisms to bring your best students into that field and kind of really attract them in it.
Absolutely.
And then you find it's kind of self-propagating, it gets its own momentum and its own life.
Yeah, so one last message, to everybody is, if you are young people, listen to this, seminar, consider Imperial, as your career, because absolutely brilliant people in Imperial. I love all of them, and that's one of the best place to be trained as synthetic biologist. Okay, so now the main speaker of today with a longer introduction, and Professor Ahmed Badran is an assistant professor, just brand new professor starting probably 8 months ago in the Department of Chemistry at the Scripps Research Institute, and one of the also famous institute for research. His work aims to prove and engineer the most fundamental biomolecules and genetic circuits in living cell, and to develop next generation solutions to long-standing global issues in healthcare and, of course, climate crisis. We need to solve this problem as soon as possible. He earned his bachelor degree from University of Arizona. Subsequently, he earned his PhD in chemical biology from Harvard University under the guidance of Professor Dave Liu. We don't need any introduction about him. One of the leading scientists, developed lots of technology regarding CRISPR system, also directed evolution, leading the development and application of rapid method for continuous directed evolution under his guidance. And following that, Dr. Badran basically was a principal investigator and fellow of the Broad Institute of MIT and Harvard University, where his independent lab, developed new technology to reprogram protein translation. He has earned several distinctions for his undergraduate and graduate research, including the Arnold and Mabel Beckman Scholarship. That's one of the most competitive fellowship or scholarship. Of course, NSF Graduate Research Fellowship, the Harvard Graduate School of Art and Science Merit Fellowship, and the National Institute of Health Directors Early Independence Award. That's another very competitive award, but he got many of them. He's very young, but I feel like I'm introducing one of the established professor. That means he's a truly rising star. And if you are, letter writer for the tenure, you should, write one of the strongest letter. I believe so. And it's absolutely my honor to host him today. So Ahmed, please take it away. It's all yours now. And thank you so much for your joining us.
Yeah, thank you for that introduction, Moon. I think that maybe the longest introduction I've ever received is also the most embellished. But I really appreciate it, and also the invitation to be here with you today to give you a little bit of insight into some of the work that we're excited about doing. So let me share my screen here. So as Moon said, my lab is really interested in a lot of these very fundamental processes in biology. And what I'll tell you about today is some of our prior and also potentially some ongoing work on trying to understand really the basis of making proteins in cells, how cells translate proteins, and how we can manipulate translation to not only understand how it works, but also to potentially develop new functions. And our motivation for this is really just from a natural observation that biology is incredibly diverse, right? I like to start with this picture just to highlight all the different types of animals that one can get, all starting from the same basis, the same genetic code, the same building blocks and biomolecular machines. And really, as chemical biologists and also synthetic biologists, there's been this sentiment that the vast diversity of functions and biomolecules in nature all give rise to very different functions, as shown here by this really beautiful image of a number of different microbes growing on agar plates. But if we can potentially develop new functions or new building blocks, and this has, for example, been shown in the context of non-canonical amino acid mutagenesis, can we potentially use this insight to develop new to nature functions? And these could be really any sort of function that a researcher can think about that maybe improve on existing functions within living cells or drive new activities altogether. And really for us, the way to achieve this by looking at how biology develops these functions is really to be able to rein in the ability of cells to translate biomolecules, either to do it more efficiently or to create new themes or new sequences or new chemical capabilities within that apparatus. And from the get-go, one of the things that we, of course, noted, and many others in this field have noticed, is that translation in the protein synthesis machinery in cells is incredibly complex. And so realistically, if we wanted to manipulate how, say, ribosomes translate proteins or the sequences of proteins that could be made going beyond the canonical 20 amino acids or so, we would potentially have to manipulate all of the different factors that you see here on the screen, including mRNA, the various initiation, elongation, and release factors, tRNAs and synthetases, and of course, the large macromolecular complex that is the ribosome that translates mRNA into protein. And really, this can be a daunting task, because if we were to put ourselves in a situation where we have to manipulate all of these biomolecules and potentially even co-evolve them or co-engineer them for new functions, that is a particularly big ask. And so we need to be able to develop technologies that really allow us or provide us with a robust handle to be able to engineer and evolve these systems. And so one of the first questions that we really began asking in this framework is, how do ribosomes work in living cells, and what types of decisions do ribosomes actually regulate in the context of living organisms? And one of the first observations that really drew our attention, this is work that has been led by Terry Hua here at UCSD in San Diego, alongside many others, that showcases that translation in living cells is potentially a rate-limiting step in how quickly cells can grow. And the way that this has been delineated is by comparing the content of ribosomes in cells, indicated by this RNA protein ratio, to the growth rate of, in this case, E. coli cells. And you can see this very nice linear correlation. And you can draw similar correlations by comparing the elongation rate to growth rate in E. coli as well. And one of the fundamental questions has been, can we enable cells to grow at faster rates, in particular by influencing how ribosomes affect translation in living cells? And some of these data might immediately suggest that one way to do that is, for example, to increase growth rate here by increasing the RNA protein ratio or the number of ribosomes in living cells. Of course, cells already commit a significant amount of resources to creating ribosomes. And so this is shown in this graph where what you're visualizing is the energy allocation to any particular stage of metabolism in living cells. And you can see the largest chunk is really just devoted to protein translation and, in fact, to making ribosomes. And from computational analyses, what we might predict would happen is that if cells overcommit to producing ribosomes in an effort to enhance growth rate, that eventually the number of ribosomes that is made in the cells is much larger than what is needed to sustain survival, and importantly, overcommits resources to building ribosomes rather than translating the products of many of the genes in living cells. And so you get this predicted relationship where higher ribosome content results in slower growth rate. And so one of the questions, and it's a very simple question that we asked, is can translation be physically optimized to operate at higher catalytic efficiency? And so if that were true, then the cell could commit the same amount of resources to creating ribosomes, but each ribosome would translate faster. And then we could really ask the question, can cells grow faster using a higher or faster translation rate? And importantly, do different ribosomes provide access to different variants that could endow cells with higher translation rates? So does the starting point for many of these engineering or evolution pipelines matter? So to really get the ball rolling on this project, we had to be able to create different ribosomes all in the context of similar or related experimental conditions. And historically, the way that this has been done is by first deriving ribosomal components. These are often ribosomal RNAs that you can see here from various microbes that are derived from the environment or used for biomedical research, and introducing them onto plasmids. And in each case, these plasmids encode the 3 structural RNAs that make up the ribosome: 16S, 23S, and 5S ribosomal RNA. And upon introducing these into E. coli, one could ask, can they assemble into ribosomes and then potentially use that to explore new functions? In E. coli, this is made readily accessible by using specifically engineered strains such as SQ171 that have been deleted for all ribosomal RNA operons in the genome. And this deficiency is complemented by a plasmid-borne version of one of these alleles, in this case RNC, alongside a counter-selectable marker, SACB. And so what researchers can do is they can combine both of these repertoire to create, momentarily at least, these mosaic organisms that encode ribosomal RNAs and therefore potentially ribosomes derived from multiple different organisms simultaneously. Upon depletion of the original resident plasmid using sucrose growth, which kills all cells that bear this SACB cassette, if bacteria survive, then they should be sustained by this heterologous ribosomal RNA, and this then provides an avenue to explore and evolve and engineer these ribosomes, all within the context of E. coli. Of course, we can extrapolate this to many other ribosomal RNAs and begin to explore phylogenetically related relationships between ribosomes from various heterologous organisms. Now, importantly, this mechanism has a number of advantages. In particular, it gets around studying ribosomes in the context of natural organisms, and so it overcomes things like potential different culturing constraints or environmental conditions that are required for certain microbes, but not others. Importantly, it also allows us to explore ribosomes from organisms that might be dangerous to grow in a laboratory, for example, highly drug-resistant microbes. And so collectively, it's a really enabling technology to allow us to study ribosomes without many of the different bottlenecks that are typically imposed by microbes in the laboratory. So in my lab, Natalie and Renan began by exploring this by first using ribosomal RNAs from a number of different organisms that you can see here. And importantly, and this will continue to come back throughout the presentation, these are ribosomal RNAs that are increasingly divergent in sequence identity from the counterpart in E. coli. And they asked the simple question, if they add this to SQ171 cells, can they get functional complementation? And what we very quickly found is that only ribosomal RNAs that are very homologous to E. coli, very similar in sequence, can you get functional complementation. And as we get increasingly divergent, this complementation goes away. Now, what we recognized, and this was work that was primarily led by Natalie Kolber in my lab, she recognized that these structural RNAs that make up the ribosome, like in the 16, 23, and 5S, are incredibly well conserved. And you can see this here in this graph, the color coded by the different elements. Conversely, the elements that are in between, the so-called intergenic sequences, can be incredibly divergent between ribosomal RNAs from different organisms. And what we hypothesized was that these sequences, which are known to fold into defined structures that are processed by living cells to liberate the structural RNAs, could be a major bottleneck for their processing in the context of E. coli. And so perhaps by taking E. coli-derived intergenic sequences or processing signals and introducing them into heterologous ribosomal RNAs, we could enable the production of these functional heterologous ribosomes in the context of E. coli. Very quickly, this is fortunately what we found to be the case. So you can see here from these wild-type ribosomal RNAs versus these engineered processing signals, sequences, we can now recover activity or complementation from Pseudomonas aeruginosa-derived ribosomal RNAs. And we can follow the complementation of these ribosomes in E. coli cells just by growing them kinetically and following the doubling time, as you can see in some of these curves. But this is a particularly large ask for many of these heterologous ribosomes. So not only do these ribosomes have to functionally assemble in E. coli cells, but they also have to carry out the translation of the entire proteome, as well as any sort of supplementary or moonlighting functions that these ribosomes may natively have in E. coli. And so we really needed a mechanism to be able to exclusively monitor the active protein translation, which would allow us to have much greater degrees of control over these heterologous ribosomes. To achieve this, we turned to a technology that was pioneered and developed by Devor and Jason Chin and many others called orthogonal translation. And the mechanism at play here is leveraging the way that ribosomal RNAs ribosomes initiate translation in microbial cells by direct interaction between complementary sequences in the mRNA and the ribosomal RNA. And so researchers can modify both of these sequences such that they're complementary to one another, but different from the starting and the wild type sequences, and in doing so create orthogonal ribosome mRNA pairs. And this is schematically shown here. Importantly, wild-type ribosomes cannot translate transcripts that bear this orthogonal ribosome binding site, and engineered ribosomes cannot translate host open reading frames. So how well does this work for our engineered and heterologous ribosomes? So this is just reminding you of what our complementation-based pipeline looks like. When we now transfer these ribosomal RNAs to orthogonal translation, we can now accurately report on the activities of these heterologous ribosomes, importantly now correlating them with activity levels of E. coli-derived ribosomal RNAs. And upon interchanging these signaling sequences or intergenic sequences, we can now see that in many cases, these activity levels rise quite dramatically. And you can see this improvement, for example, in Pseudomonas aeruginosa-derived ribosomal RNAs here. This was very exciting for us to see because it provided us with a framework to now begin to explore ribosomal RNAs from many different organisms, all within the context of wild-type E. coli cells. So at this time, we went to every lab that we could find that could give us ribosomal RNAs or microbes that might be interesting to explore in the context of E. coli. And our hunt basically yielded about 35 different organisms of ribosomal RNAs, and you can see them here phylogenetically shown where E. coli is at the very bottom of this tree. And we find some very interesting and perhaps somewhat predictable outcomes from this analysis using orthogonal translation. The first, which is what we call class 1 ribosomal RNAs, and these are RNAs derived from organisms that are phylogenetically very similar to E. coli, show very high levels of activity in orthogonal translation, and importantly, don't show any dramatic improvements upon interchanging these the intergenic sequences with E. coli counterparts, suggesting that processing was actually not as significant bottleneck for many of these ribosomal RNAs. Now, in this class 2 ribosomal RNAs, where we hit this sweet spot, where these ribosomes can function quite effectively in the context of E. coli, but importantly are restricted by their natural processing signals, which are not recognized by E. coli RNases. So when we engineer these to bear the E. coli counterparts, you can see in many cases they have a dramatic increase in activity, in some cases reaching E. coli levels, which in this graph are 100%. And finally, and again, unsurprisingly, once you get even more phylogenetically divergent, these activity levels drop quite precipitously to virtually undetectable levels, suggesting that there are potentially other elements that need to be improved or supplemented in the system to enhance the activity of these heterologous ribosomes. Now, importantly, both the orthogonal translation mechanism and the complementation-based mechanism, which you can see on these different axes, show a very strong and linear relationship to one another, suggesting that even though these ribosomes in the orthogonal system are not supporting the viability or the growth of E. coli, they are in fact reflecting the catalytic and translational capabilities within living cells, but importantly allow us to do these assays in a fraction of the time. So emboldened really by these results, we began to explore how we could further optimize activities of heterologous ribosomes in the context of E. coli, especially for ribosomal RNAs that you can see here that are reasonably active, that are quite detectable in our system, but are not at E. coli levels of activity. So having been satisfied by our optimizations of the ribosomal RNA processing, we next turn to the other structural element of the ribosome, namely the ribosomal proteins. Now, the ribosome, depending on which microbe it comes from, can have a slightly different number of R proteins that are associated with it. On average, it's between 50 and 55 R proteins, and these interact with the large subunit or the small subunit of the ribosome. In general, these R proteins are organized in operons that we can very easily find across really any microbe that we look at. These R proteins are, the majority of R proteins are organized in 5 wild-type operons that are named here for historical reasons, with the majority kind of scattered throughout the rest of the genome, as you can see here schematically. And so our approach was to subclone each of these operons, the 5 natural ones, as well as collect all of the remaining R proteins that you can see around this genome onto 2 artificial operons that we call AO1 and AO2 that are enriched in small subunit R proteins or large subunit R proteins, respectively. And then we began asking the question, if we were to overexpress these R proteins alongside their cognate heterologous ribosomal RNAs, can we further improve the function of these engineered ribosomes? And to explore this initially, we used Acinetobacter baumannii ribosomes that you can see here, which are quite functional and detectable, but still have some room to clearly grow. So initially, by testing this R protein repertoire from A. baumannii, we were excited to see that AO1 showed a dramatic increase in activity of these ribosomal RNAs, now reaching 100% or E. coli levels, E. coli ribosome levels of activity. Importantly, when we deconstruct this plasmid, which we had engineered, this is not a naturally existing operon, we found that 2 R proteins, namely S20 and S16, were responsible largely for the activity improvements that we saw, with the remaining R proteins really not contributing. This was particularly interesting because S20 and S16 are actually known to orchestrate some of the earliest interactions with the 5' end of the 16S ribosomal RNA. This is what ultimately becomes the small subunit of the ribosome, and dictate conformational changes, as was beautifully shown by Sarah Woodson's group, in elements of the ribosomal RNA that enable downstream 30S or small subunit assembly, and importantly, obviate the creation of unstable intermediates in the early stages of ribosome construction and bioassembly. And this was interesting because using this very empirical method, we had determined that one of these R proteins, or 2 of these R proteins, were effectively pushing the equilibrium towards assembled ribosomes in E. coli cells. And so this provided a mechanism to really begin to explore how ribosome biogenesis can be affected for many of these heterologous ribosomal RNAs. But importantly, do these ideals hold beyond A. baumannii? So if we were to go even more phylogenetically divergent, would we still be able to recover activities in these cells? So we carried out another round of this optimization and identified 2 additional R proteins, namely S1 and S15. And you can see here in this phylogenetically dependent manner, we can further increase the activity levels, in some cases quite extensively, of phylogenetically divergent ribosomal RNAs from E. coli. This analysis also showcased one of the most important elements that interact with the ribosomal RNA, namely S15, which orchestrates a massive conformational change in this helix in the small subunit, namely helix 21, to effectively see the interactions for all the downstream R proteins that you can see in this figure. So again, we're really starting to explore some of these early stages in ribosome biogenesis using this heterologous and orthogonal ribosome assembly mechanism. And importantly, we're seeing this clearly phylogenetically dependent outcome for many of these assays. So the question then that we asked is, rather than empirically deriving phylogenetically motivated results, such as the ones that you're seeing here, can we flip this around and use phylogenetic differences between E. coli and a target organism to nominate R proteins that would improve the function of even more divergent rRNAs. So we went back to our library of ribosomal RNAs and mined the genomes from which they come for R proteins and compared their sequence identity to the E. coli counterparts, as you can see here. And so one of the striking things from this analysis is that proteins that we had empirically identified that improve the function of these heterologous ribosomes, such as S20 and S16 and S15, were among the more divergent R proteins across many of these organisms, suggesting that we're already kind of on the right track. So using this analysis, we nominated a completely different repertoire of R proteins that you can see here in dark blue to enable the activity of much more divergent rRNAs that you can see here from the blue arrows. And using this analysis, we can now improve the function of even gram-positive ribosomal RNAs in the context of the gram-negative E. coli for the first time now providing access to such divergent ribosomes in a very different cell. This was a very exciting study for us because it really allowed us to explore how to assemble these heterologous ribosomes in the context of E. coli and provided us with a very vast library of building blocks that we can now use to explore the question that I started with at the very beginning, which is, can we begin to affect translation rates in living cells and explore their relationship with how quickly a cell might grow? Now, to be able to do this, we need very powerful methods of evolving biomolecules in particular because the ribosome is a massive complex. And so being able to evolve it would potentially require significant investment of time and resources. Now, fortunately, as a graduate student, I had spent a lot of time working on a mechanism that allows us to explore directed evolution in a very rapid timescale called phageassisted continuous evolution. And the basis behind PACE is using a bacteriophage life cycle to rapidly evolve virtually any biomolecule of interest. So I'll briefly walk you through how this works here. In the first step, we take a general M13 bacteriophage and engineer its genome to encode an evolving gene of interest in place of an essential bacteriophage gene called gene 3. Now, gene 3, when translated into P3, regulates both cell entry and viral export from of the progeny viruses from the infected cell. So it's a very important protein in the viral life cycle. And without it, bacteriophage are non-infectious or potentially not produced at all. And our basis for then evolving this biomolecule is creating a plasmid, which we call the accessory plasmid, or AP, that somehow affects a genetic circuit in this plasmid to affect the production of P3, and in doing so, complementing the deficiency from what we call the selection phage, or the SP. Now, this gives us a very strong selection mechanism, but we also need mechanisms of introducing mutations into the evolving gene of interest. And to do that, we overexpress a number of mutagenic proteins or mutator proteins that increase the mutation rate over 300,000-fold in the context of living E. coli cells. So when we start a PACE experiment, we then engineer these host cells that bear the accessory plasmid, or AP, and mutagenesis plasmid, or MP. And upon infection, mutations are accumulated across all constructs, including the AP, MP, and SP. And the selection phage, if it encodes a functional biomolecule, will result in tripping the circuit on the AP, the production of P3, and the production, therefore, of infectious progeny phage. Conversely, if that biomolecule is inactive or not active enough, the amount of P3 that is produced is much lower, and therefore the produced progeny are non-infectious. And we can continue to iterate this process in what we call the lagoon, which is a constant volume vessel wherein we continuously import or add new host cells that have been engineered with these two plasmids, and constantly outflow material by controlling the flow rate in the lagoon such that it is faster than the doubling time of E. coli, but slower than the replication time of the bacteriophage, so that on average, the only thing that accumulates in the lagoon is the SP, and the mutations that allow the SP to overcome the selection will largely accumulate in the evolving gene of interest. Now, this mechanism enhances the rate of directed evolution approaches by on the order of 100-fold, and this permits us now to do 50 rounds of directed evolution or more, depending on the selection mechanism. Now, this approach has been applied extensively to various protein activities, including polymerases, protein-protein interactions, proteases, and a lot of work that has been done in the Lou lab now has really extended this to virtually any biomolecule that could be explored. We were particularly interested in really extending it to the evolution of ribosomal RNAs. And so we coupled, and this was now work that was led by Fan and Sinisha in my group, we coupled orthogonal translation to M13 bacteriophage propagation by introducing our orthogonal ribosomal RNAs on the SP and generating an AP that encoded the orthogonal RBS upstream of this M13 gene 3. And so this, in doing so, we would couple phage-borne or SP-borne ribosomal RNAs to the production of P3. And this would allow us then to constantly evolve these ribosomes at pace. So in the first experiments, we first introduced a number of deleterious point mutations in the phage-borne or SP-borne ribosomal RNAs. And you can see here that these are quite deleterious. So we compare the input titer, which is here shown at 10 to the 5th, to the amount of phage that's produced upon infecting cells bearing the AP. So if it's functional, we see dramatic enrichment, but if it's non-functional, we see this dramatic de-enrichment of phage titer. And readily in PACE, we can very quickly recover the identities of point mutations at each of these positions to the natural nucleobase in very, very short timescale. So in this case, we can revert the identity of even this triple mutant in less than 5 hours of orthogonal ribosome PACE or ORibo PACE. And this really showcased to us that we can now begin to evolve potentially novel and exciting activities in engineered ribosomes. So to begin to explore this, we evolved 3 different ribosomes. Of course, the E. coli ribosome, which has been extensively characterized, but also ribosomal RNAs from Pseudomonas aeruginosa and Vibrio cholera to explore kind of the dependence of the outcome on the starting point. We carried out these selections over the course of a number, about 200 generations in PACE, where importantly, we varied the flow rate in the lagoon, and this allows us to moderately affect the selection stringency, but also by manipulating the AP to make the selection more difficult. So in the first segment, we exclusively evolved these ribosomal RNAs on an easy selection to enable neutral drift and the accumulation of mutations. And in the second segment, we reduced the concentration of this mRNA, which potentially might influence the evolution of ribosomes towards improved activities. We later developed technologies that allow us to artificially elongate the mRNA so that the ribosome, which is being now subjected to a kinetic selection, has to progress through the course of the whole elongated gene to enable phage propagation. And this would potentially allow for the evolution of faster ribosomes. We also explored kind of the path dependence of these evolutions by going from our neutral drift stage directly to this longer mRNA. Once we finished carrying out these experiments, one of the first things that we noticed is that these ribosomes seem to evolve along different trajectories in some cases. And in particular, we noted that Vibrio cholera-derived ribosomal RNAs, as you can see here in purple, seem to accumulate mutations at much faster rates than both E. coli and Pseudomonas-derived ribosomes in the context of ORibo PACE. This potentially has bearing for the evolvability of different ribosomal RNAs, and this is something that we're still actively exploring. So over the course of these 200 generations, what actually happens to these ribosomes? To answer this question, we developed a broad repertoire of assays that allow us to assess the catalytic and protein output of many of these ribosomes. And I'll briefly just show some of the data here. First, by analyzing the total protein output, we found that in many cases, these ribosomes show moderate to, in some cases, quite significant improvement in the amount of protein that they make. Again, Vibrio cholera ribosomes seem to do quite better than E. coli, which seems to do better than Pseudomonas. And this theme seems to track throughout all the experiments that we did. We also found that the R proteins that we had identified in our previous studies seem to complement evolved ribosomal RNAs that were derived from ORibo PACE. And so we can get this additive effect by providing these necessary proteins even after evolution. And surprisingly, when we explored functions that we had no selection pressure for in the context of PACE, namely non-canonical amino acid incorporation, we found that many of these ribosomes showed dramatically improved outputs, in some cases reaching up to 10% of the conventional E. coli orthogonal translation technologies that have been established in the field. And so this might suggest that there is an interplay between the speed of translation and the efficiency of genetic code expansion efforts, and this is something that we're actively exploring now as well. So how did these ribosomes achieve these improvements? First, we looked at the types of mutations that were accumulated across ribosomal RNAs from these various organisms. And as you can see, even though there are commonalities in the mutations that show up, they're not identical trajectories, and they seem to depend on the starting point that was used for many of these evolutions. Now, most importantly, these mutations all seem to accumulate near very specific elements in the ribosome, namely the decoding center or parts of the ribosome that are responsible for checking the tRNA-mRNA interaction at some of the early stages of translation elongation. And you can see these here kind of color-coded near the tRNA that's in the A site. There are other mutations that seem to show up elsewhere that we still don't understand exactly what they're doing, but the mutations that show up near these decoding centers seem to be very strongly selected for across many different ribosomal RNA trajectories. This was both exciting and a little bit scary for us, because not only would this be a step that would be implicated in faster translation, if the ribosome could select tRNAs faster, then that would make sense, but also because the decoding center is involved in maintaining the fidelity of translation. This might suggest that these evolved ribosomes not only translate faster, but they do so with reduced fidelity. So to really explore this, we went back to our complementation-based method and reintroduced in these ribosomal RNAs using now a wild-type anti-RBS so that they could sustain the viability of our SQ171 cells, and then transform them with plasmids that overproduce a model protein, in this case GFP, that we could use to quantify the mistranslation rates in these cells very accurately. And surprisingly, what we find is that by comparing the wild type or the starting point mistranslation rates that you can see here in these filled circles to the evolved variants, either from E. coli or from Vibrio cholera, we didn't see a dramatic increase in mistranslation efficiency that might suggest that these ribosomes had in fact become less or became more error prone than their starting parental ribosomes. Excitingly, though, when we used mechanisms that allow us to explore the efficiency and the speed of translation in living cells, and this we achieved using incorporation of the methionine analog azetohomoalanine, we find that in many cases these evolved ribosomal RNAs translate at much faster rates, reaching up to 3 to 4 times faster than their natural or parental ribosomal RNAs. Interestingly, however, when these ribosomal RNAs are introduced into SQ171 cells, if they are indeed faster, which we've corroborated now by a number of different methods, we might expect these cells to grow faster if the originally described models were in fact correct. Unfortunately, what we see is something very different. Either the cells grow at the same rate as the starting parental strains, or they grow slightly slower. And so we're still trying to understand what this means, but it might suggest that even though the ribosome plays a significant role in dictating the cellular growth rate, it may not be the only factor that influences this. And so potentially other biomolecules may need to be engineered or evolved to access improved growth rates. So with that, I'll just briefly summarize some of the work that we've done today. So we've done a lot of work on heterologous ribosome assembly in the context of E. coli and established guidelines now that allow us to mine ribosomal RNAs and Rproteins from any organism that is moderately phylogenetically divergent from E. coli, and really establish the function of the ribosome in cells that are genetically tractable. We've used this mechanism to now evolve ribosomal RNAs towards new activities and explore the relationship between cell fitness, protein yield, and translation elongation rates. We've also explored the essentiality of elements within the ribosome itself, where we've been able to show that even though there are sequences that are incredibly well conserved in the ribosomal RNA, in many cases they are dispensable for the act of translation. And this has implications for potentially how the ribosome may have evolved, or the ribosome that existed in the last universal common ancestor. And finally, in work that I didn't have the opportunity to talk about today, we've explored the evolution of other RNAs in living cells, in this case, tRNAs that decode quadruplet codons that may have tremendous utility in genetic code expansion efforts in the future. And we've shown that using PACE, we can readily evolve these tRNAs to be very efficient, mutually orthogonal, and importantly, also quite processive. So we're actively exploring all of these efforts, both to understand how we could further manipulate the translation in living cells, but also to potentially afford new functions in translation. So with that, I'd like to thank all the people that did the work. I tried to highlight the various folks that led the studies as I went through the presentation, but of course, the whole lab has contributed to all the work that I showed here today. All our significant contributions, I would say, were enabled by fantastic collaborators, as well as all the funding sources that you see here on the slide. And finally, also echoing sentiments from Geoff earlier, if you're interested in any of this work or some of our ongoing work in carbon fixation or creation of new genetic codes, we're actively hiring, hopefully as quickly as we possibly can. So please feel free to reach out at any time. Thank you, and thanks again, Moon, for the invitation. It's been a pleasure, and I'd be happy to take any questions.
So amazing talk, and also competition between two university or institute. That's amazing. So, Dr. Baldwin, do you have any comment or question before I, invite some question from audience, or I'm asking question? Do you have anything?
I'll start, Tae-Sok.
Sure.
Fantastic talk, Ahmed. Thank you. I just, in terms of the PACE system, always strikes me as being quite complicated. So I just sort of wondered what's the hardest thing about working with that as a system?
Yeah, I would say early on in the development, the major bottleneck was really building it. So it's liquid handling and peristaltic pumps and tubing, and in some cases, even working in a warm room, which, if you're in a very hot and humid area, that can be quite frustrating. But more recently, there's been a lot of innovations in this space to make it a lot more accessible. So there's versions of this that are now compatible with liquid handling robots, such as the ones that you highlighted earlier today. So that could dramatically make it easier to access. But also there are, I would say, manual versions of PACE now called PANTS, where you can effectively passage your bacteriophage on cells that you grow in a culture. And that also makes it a lot more accessible. Really, from a molecular biology standpoint, the biggest bottleneck was selection development. And this applies across all directed evolution schemes. I would say now there is such a large repertoire of selections that potentially one could plug into, where you exchange your biomolecule of interest for the one that was described in the original study. And my expectation is that you'd be very quickly off to the races. If there were a new selection that one wanted to pursue in the context of PACE, that I think would be a pretty steep learning curve to put that together. Yeah.
And I suppose the other kind of corollary to that is that, in thinking about directed evolution, I mean, the classic way of doing PACE in a sort of single vessel kind of bioreactor with the dilution, whatever, effectively you have whatever evolutionary trajectory goes on in that vessel, right? Whereas I kind of really like the idea of doing it in a 96 well plate when you then have 96 evolution trajectories.
Yeah.
And I just wondered, do you think that makes any difference?
So hard to tell. I mean, as is the case with most directed evolution pipelines, it really just depends on so many different things. But beautiful work that's come out of Cheng Lu's lab and also more recently Kevin Esvelt's lab really shows that in some cases the evolution trajectory can be different. And you can start to see these now if you have so many replicates that you begin to capitalize on very, very rare events. It may not necessarily influence whether or not the selection works. That I think there's tuning that one could do to really inform how to get from point A to point B. But I would say the replicates and informing the path dependence could become instructive from a basic biology standpoint to understand more about the protein rather than to affect the final endpoint.
Cool, thank you.
Okay, thank you. So let me check the audience question quickly. I do not see Q&A. I do not see chat. That means your talk was perfect. I have one question. So we originally or briefly discussed before the seminar, but I also want to ask one more time. So could you share your insight into two approaches? Of course, a prediction guided approach using AlphaFold, whatever algorithm out there, versus directive evolution. You kind of outlined beautifully today. That is of course complementary, but for the junior people, young people, they still kind of wonder which one or how you combine those things together. That is one of the biggest kind of question young people might ask. So I just want to ask for them, and then I just want to hear your perspective, because that discussion not recorded earlier.
Yeah, I guess for us, I can tell you that we use all of these approaches very regularly. In general, we found the predictive approaches to be really enabling where there's virtually no information, no biochemical information or no structural information that's known about the protein, but there's some observations that might suggest, protein has a specific function or might be modified to have that a specific function. In some cases, it's actually nominated starting points for us that have been quite successful at evolving in a researcher-dictated manner in PACE or in other types of selections. So I would say, I don't expect that any one or the other is going to dominate. As we spoke earlier, directed evolution is really much better at finding the weird solutions, which are often easier and less predictable because they're too far away from where we might conventionally look for different activities like active sites. But I think between the two of them, we could probably solve many, many problems that we may want to address. So I would say, really being comfortable with different directed evolution approaches as well as these predictive approaches probably goes hand in hand and will be a successful mechanism for anyone doing synthetic biology in the future.
Yeah, so of course, my challenging question you answered beautifully. So that's wonderful. So right now, my time right now I talking, about the UK time. So let me check my UK time. That is the 5:10. So we need to probably close. So thank all for joining and staying today. We'll meet again next week on June 2nd. Thursday, the same time, the same Zoom link, but I will be joining from Germany after my 4th seminar during this crazy trip at Max Planck Institute for Chemical Ecology. Interestingly, we'll have Professor Ahmed Badran from University of Toronto, Canada, and Professor Nathan Crook from NC State University, making all three of us joining from different countries, actually 3 different countries for the first time, I believe. So as usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty face. And thank you so much for joining us. And I stop recording. Let me stop. Okay.